stop and think about how your body knows which genes to turn on and which ones to just leave off? It's a huge question, right? And the answer, well, it's not just buried in the DNA code. It's actually all about the intricate three-dimensional shape of molecules. So today, we're going to dive into how that fundamental shape literally writes the rules for the very function of life. All right, let's start by zooming way, way in, right inside the nucleus of one of your cells, because that's where we find one of the biggest, most fundamental mysteries in all of biology.
I mean, think about it. You've got trillions of cells, and every single one has the same instruction manual, the same DNA. So how on earth does a liver cell know to be a liver cell and not, say, a brain cell? What's the conductor of this absolutely incredible biological orchestra?
Well, the answer comes down to these really specialized proteins called gene regulatory proteins. And the best way to think about them is like tiny molecular keys. Each one is designed with incredible precision to fit into a very specific lock on your DNA. And when it clicks into place, it can either turn a gene on or switch it completely off.
Okay, so if this whole thing is about a perfect fit, a key finding its one and only lock, then the first big clue has to be their physical structure, right? It all comes down to one thing. The secret is shape.
Now, the key word here is complementary. And we're not talking about a loose fit, no way. This is more like an exact molecular handshake. Every little bump on the surface of the protein fits perfectly into the grooves of the DNA, and all the chemical charges have to line up just right. It's incredibly precise.
And what's really amazing is how they do this. The protein doesn't even need to unzip the DNA helix. Instead, it just reads the outside. It scans for a unique pattern of atoms that are exposed in what's called the major groove of the DNA. See, each base pair, the A, T, C, and G, presents a slightly different chemical face to the outside, and these proteins have evolved to be expert face readers.
But, this just pushes the question one level deeper, doesn't it? We know shape is everything, but what gives these molecules their incredibly precise shapes in the first place? How does a simple chain of amino acids know how to fold up into the exact protein key it needs to be? For that answer, we actually have to take a little detour from biology and dive into the fundamental rules of chemistry.
And what you'll find is that there's this surprisingly simple, almost elegant set of rules that governs the geometry of all molecules. I'm talking everything from a tiny water molecule all the way up to the most complex protein in your body.
Okay, it's called the valence shell electron pair repulsion theory. Yeah, I know, it's a mouthful. We just call it VSEPR theory. But trust me, even though the name sounds complicated, the idea behind it is something you can totally get. So here's the core principle, and it's super simple. Electron groups around a central atom repel each other. They're all negatively charged, right? So they just want to get as far away from each other as possible. Think about it like this. If you tie a bunch of balloons together at their knots, what happens? They automatically push each other away and spread out to take up the most space. That's exactly what electron groups do around an atom. But here's a little twist. Not all of these electron groups push with the same force. Some are, well, they're bigger bullies than others. The biggest bullies of the molecular world are what we call lone pairs. These are pairs of electrons that aren't tied up in a bond. They take up a lot more space and they have the strongest repulsive force, basically shutting all the other electron groups further away. And this idea is where things get really, really interesting, because it means that one of the most important things determining a molecule's final, visible shape is actually the electrons you can't see. It's like an invisible influence. Okay, let's look at a molecule we all know and love. Water, H2O. You've got an oxygen in the middle and two hydrogens on the sides. So why isn't it just a straight line, HOH? Seems like it should be, right? Well, the answer is, you guessed it, those invisible, bossy lone pairs we just talked about. So to really get this, we have to make a super important distinction between two different kinds of geometry. First, there's the electron geometry. That's the 3D arrangement of everything, all the electron groups, the bonds, and the lone pairs. Then there's the molecular geometry, and that's the shape we actually see. It's just the final arrangement of the atoms themselves, after the invisible lone pairs have done their pushing. Let's break it down for our water molecule. The central oxygen atom has 4 distinct groups of electrons buzzing around it.
You've got the 2 bonds connecting to the hydrogens, and you've got those 2 lone pairs. So that's 4 electron domains in total. And what do we say 4 groups do? They arrange themselves to be as far apart as possible, which forms a shape called a tetrahedron. So that's water's electron geometry.
But remember, when we look at the final shape of the molecule, the molecular geometry, we don't see those lone pairs. They're invisible. We only see what they did. Those 2 powerful lone pairs sitting on top of the oxygen literally push the 2 hydrogen atoms down and closer together. And what you get? A bent shape. It's the perfect example of how the invisible electrons dictate the final visible structure. And this pattern isn't just for water. It's a rule. Look at other molecules that also have 4 electron domains. You take methane, CH4. It has 4 bonds, but 0 lone pairs. So its molecular geometry is the same as its electron geometry, a perfect tetrahedral. Now look at ammonia, NH3. It has 1 lone pair. That one bully pushes the 3 hydrogens down into a shape called trigonal pyramidal. And then you have water with 2 lone pairs, which gets squashed even more into that bent shape. See the pattern? They all start with the same basic tetrahedral electron arrangement, but adding invisible lone pairs completely changes their final molecular shape. All right, so we've just been deep in the weeds of chemistry, right? We've seen these simple, powerful rules. Now it's time to zoom back out and connect all of this back to that huge, complex world of biology where we started this whole journey. So let's recap the whole story. It all starts with the most basic principle you can imagine: simple electron repulsion. That simple push, guided by those VSEPR rules, is what creates these incredibly complex and specific 3D shapes. And that shape, well, that shape determines everything. It's not just a minor detail, it's the whole ballgame. In biology, shape is function, period. So here is the big, mind-blowing takeaway. The exact same fundamental VSEPR rules that make a simple water molecule bent are the very same rules that allow a complex protein to fold into a perfect helix-turn-helix motif, the exact shape it needs to read a specific gene on your DNA. That shape allows it to slide perfectly into the DNA groove and act as the on-off switch. From the smallest, simplest molecule to the most complex machinery of life, the rules are exactly the same. And that leaves us with one last, kind of cool thought to chew on. That whole lock and key idea we've been using, it's a great model, but it's a little bit of a simplification. The reality is, molecules like proteins aren't perfectly rigid statues. They're constantly wiggling and vibrating in a range of possible shapes. So maybe binding isn't a single click of a key in a lock, but more like a process of selecting the best fitting shape out of many possibilities. It makes you wonder, right? Maybe life isn't a perfect rigid machine, but something much more fluid, a constant dynamic negotiation happening at the molecular level every single second.